into time. So with that, we will have our first speaker who we recorded about a month ago, the O'Connor and Kushner Labs, join for a Q&A session. So Anusha, if you want to take over and start that presentation.
Yeah. Please let me know if you cannot hear it.
They can mute themselves. Excellent. So it's my pleasure to introduce one of our pilot study awardees for MGNet, Jan Luneman, and he's going to be presenting his work, Antibody Omics in Myasthenia Gravis. So all yours, Dr. Luneman.
Thank you very much, Henry. I'm very sorry that I cannot attend the meeting in person or remotely. Since before the date was fixed, we booked a flight to visit our second daughter who's doing a voluntary year in Africa. And I'm sitting on a plane to Uganda while you will meet on Good Friday in Washington, D.C. So thank you very much for inviting me and for the opportunity to have that recorded and to present the data that we obtained during a project which was supported by the MGNet network. Myasthenia gravis is a heterogeneous disease in terms of clinical presentation, in terms of age at onset, in terms of thymic pathology, and also in terms of the specificity of the antibodies involved. Most patients, they develop antibodies against the acetylcholine receptor, 85 to 90% of patients. And these antibodies, they impair neuromuscular transmission at the end plates by blocking the acetylcholine receptor, by inducing internalization of the acetylcholine receptor, or by recruiting elements of the immune system, such as complement, which ultimately can impair integrity of the muscle membrane. Now, these functions of the antibodies are mediated by, on the one hand, the FAB domain, which binds to the receptor, but on the other hand, by the FC domain, which recruits elements of the immune system and can instruct the immune system. And these elements include complement on the one hand, but also cellular elements, which bind to the FC domain of the antibodies with the FC gamma receptors. And these FC-mediated effector functions, they are regulated by a tiny structure which is buried in the so-called CH2 domain of the antibodies. This is the N-glycan. And for example, absence of glucose on this glycan is associated with a higher ability of the antibody to bind to activating Fc gamma receptors and then induce lysis of the target cells. This is, for example, why all of the newer developed immune cell depleting antibodies, for example, CD20 depleting antibodies, they lack glucose to enhance the ability to kill target cells. And these terminal sugar residues, galactose and sialic acid, they are central in mediating anti-inflammatory effects of these antibodies. So antibodies do not only bind to antigen, they can instruct the immune system, they have multiple functions and beyond just antigen binding. And we developed a platform that allows us to basically profile functional and physical chemical properties of IgG antibodies, and we call that antibodyomics or system serology. So what we're doing here, we isolate antibodies, antigen-specific antibodies from the blood of patients, in this case with myasthenia gravis. And here we use the alpha-1 subunit of the acetylcholine receptor, and then we basically take these antibodies apart. We look how good is the antigen binding, what is the binding affinity, how good are these antibodies in inducing complement activation, NK cell activation, in mediating phagocytosis. What is the affinities of these antibodies to these Fc gamma receptor, which are very important in instructing cell-mediated immunity, and how is the Fc glycan composed? And then we obtain a lot of data, then we integrate this data with clinical, demographical data, with the aim to basically serological signatures that associate with disease phenotype, disease severity, or even treatment response. And we applied this platform to different antibody-mediated diseases. One was MOGAD, which is a CNS disease mediated by antibodies specific for oligonucleotide glycoprotein. And what we could show here is that basically higher Fc gamma receptor binding, higher ability to activate NK cells, lower Fc cyanidation is associated with clinical disease activity. This was enriched in patients in which the blood was taken during a relapse as opposed to remission. And another example is rare autoimmune encephalitis, CASPA2 antibody-mediated disease. What we could show here is that basically IgG1, so the subclass IgG1-driven effector functions, are associated with higher disease severity and poorer outcome in these patients. So this is kind of a proof of principle that we can use this platform to find basically or to identify clinical serological correlates. Here we analyze samples from MG patients undergoing thymectomy. Thymus plays a central role in patients specifically with early onset myasthenia. Most of those patients show thymic focal hyperplasia. This is characterized by B-cell infiltrates. Germinal center-like structures can form. Lymphoid structures that you usually find in secondary lymphoid organs, such as lymph nodes, for example, are present in the thymus in patients with myasthenia. This is not specific for myasthenia. You can also see that occasionally in other autoimmune diseases, but it's a prominent feature specifically in early onset myasthenia. So we had the opportunity to profile the leukocyte architecture of thymi derived from patients with myasthenia very recently. That was work done by a PhD student in my lab. And what we could find here by analyzing single-cell suspensions with a single-cell RNA-seq analysis is that these cells were basically frequently colonized with thymus. That is what was known before. The frequency is around 10-fold higher than what you would see in thymic samples derived from patients not diagnosed with myasthenia. So there are a lot of B cells in there, and these B cells are specifically memory B cells, but also germinal center-like B cells and plasma cells are found in these thymi, and those cells are also clonally expanded. Specifically, memory B cells and germinal center B cells are clonally expanded in these thymi, and we know since quite some time, this was the pivotal study of John Newsom Davis group and Vincent Turk. From 1975, that when you culture thymic tissue from myasthenia patients, you can identify acetylcholine receptor specific antibodies in the supernatant cell. And this observation supported the therapeutic approach to perform thymectomies in patients with myasthenia. And the first randomized clinical trial that investigated the effects of thymectomy in myasthenia gravis was this one, published in 2016, so more than 10 years ago. started, and there was a extension trial, 2 years extension trial of the MGTX trial published a few years later. And what these trials could show is that in patients that received thymectomy plus prednisone, compared to patients who received prednisone only, that the former have a sustained improvement, and that thymectomy leads to a significant steroid-sparing effect. But interestingly, the stabilizing effects kicked in usually not before month 6 to 9 in most of the patients. So it's not an effect that occurs immediately after thymectomy. Actually, after thymectomy, myasthenia can actually worsen, but after a couple of months, the patient improve. And so we set out, and this work was done by Carla Souza, a postdoc in my lab, who left her home country, Brazil, to pursue her postdoc and tackle this project. We, in collaboration with Henry, we analyzed basically the antibody responses using this antibiotic platform from patients who participated in the MGTX trial, and we analyzed samples that were obtained at baseline, so before thymectomy were performed, and at month 36, as well as at month 6. So 3 time points. And altogether, we included 78 patients with MG, 38 who received prednisone only, and 4 who received thymectomy and prednisone therapy. And we used a binary outcome measure here, and this was clinically meaningful improvement by at least 3 points on the QMG scale. This is an outcome measure which is usually used in most of the clinical trials that have been performed in recent years. And using this binary outcome measure, we observed that 72% of patients that received thymectomy in addition to prednisone classified as responder, 61% classified as responder of those patients that received prednisone only. So we first analyzed antibody titers in these patients, comparing prednisone-treated patients and thymectomized patients. And what we observed was that the titers went down in both treatment groups, but the thymectomized patient showed an accelerated decline. That was highly significant already at month 6. And while the significance was observed in the prednisone-treated patients only at month 36. And if we then compare responders and non-responders in the prednisone and the thymectomized group, we observed that the decline was associated with a clinical response, because the patients that did not respond by 3 points on the QMG scale or more, they did not show a significant reduction of the acetylcholine receptor antibody titers. And as for the whole group, we see here that responders to thymectomy, they show an accelerated reduction in acetylcholine receptor antibody titers. This was done by immunocytochemistry with the Euroimmune kits. However, at all time points investigated, baseline month 6 and month 36, titers did not discriminate between responders and non-responders to thymectomy. So we therefore used the kind of systems immunology approach to identify features that could discriminate between responders and poor responders to thymectomy. And so we basically applied those samples to the assays that I just introduced, and then performed the PLSDA lasso analysis. And what we found was that we could distinguish responders to thymectomy from non-responders here in orange, and this is basically in pale beige. We could discriminate between the two with high accuracy. And the most discriminating features between responders and non-responders were actually related either to Fc receptor binding or to glycosylation. And this figure here shows you a fully processed FCN glycan, and you can see that the terminal sugar residues are either galactose and sialic acid, and what was enriched predominantly in the responders was the sialic acid content. We then moved on and performed univariate analysis to see whether that confirms our basically lasso-based observations. And what you see here, these in orange are thymectomized patients, and in blue are prednisone-only treated patients. And after in month 36, we found a significant reduction of glycoforms that lack terminal galactose and sialic acid in the responders, and vice versa, an increase in glycoforms that that had terminal galactose and sialic acid residues. This was at month 36. There was no difference in the prednisone-treated patients, and the difference was not observed at baseline, so it was not a pre-existing condition. It occurred after thymectomy, and for most of the glycoforms, it was not present at 6 months, with exception of the G2. So there was already a difference in the univariate analysis for the galactosylated glycans. So lack of terminal galactose, lack of terminal sialic acid is usually associated with animal models of autoimmune disease with the more pro-inflammatory functional signature of antibodies, and are frequently seen also in patients with autoimmune diseases such as lupus, for example, rheumatoid arthritis, in those patients that have flares, so higher disease activity. We also performed a linear regression, also used a linear regression model to see if that supports our findings. Here we adjusted also for age and sex, and what we could see here, this is at month 36, is that the difference we observed in the multivariate and the univariate analysis are also visible here in the linear regression model at month 36. At baseline, there are no difference in glycoform frequencies, and it's already at month 6 here also we observe decrease in G0 glycoforms in patients responding to thymectomy. So in summary, what we observed was there's an improvement, that clinical meaningful improvement is associated with the decline of acetylcholine receptor-specific IgG over time. That antibody features discriminating responders from poor responders are linked to IgG, Fc glycosylation, and that the clinical benefits strongly associated with an anti-inflammatory IgG glycosylation profile, and that indicates that intrathymic IgG glycosylation may contribute to MG pathogenesis and deserves further research. So I would like to thank all the collaborators with whom I had the pleasure of working with during the project, particularly Henry, Yannick, Gordon, and John. Special thanks to Carla, who performed all of the experience and the analysis, and of course to MGNet, who kindly supported this project. I would like to thank you for your attention. I'm happy to take any questions.
Yeah, that's fantastic. They can mute themselves. Excellent.
Okay, yeah, we had a Q&A, but now we will end and move on. I do have one more comment about the last presentation. Those samples that were used would not have existed if Story Landis, who is the head of the NINDS, did not agree to add funds to the clinical trial to collect those. And now, as you see, they're bearing fruit. Anna Punga has used those. Alex Marks has done a deep dive into the thymic specimens that were analyzed. That was also supported by the MGFA. And then Linda's lab is doing a deep dive with colleagues here at GW doing whole RNA-seq and proteomics. So they continue to bear fruit. So, again, kudos to the NIH supporting myasthenia gravis research. And Dr. Leach, it's all yours.
Hi, everyone. I'm Justin Leach. I'm assistant professor of biostatistics at the University of Alabama, Birmingham. This is a project with Chi-Chi Aban, Gary Cutter, and Michael Benatar. So next slide. So our outline, it looks like a lot, but not as much as you might think. So the first thing I'm going to talk about is just how is it that rescue therapy complicates statistical analysis, in particular of MG trials. I'm going to attempt to convince you that it's something you should be thinking about if you're not already. I'll very briefly get into something called estimands. This is essentially a fancy statistical term for the treatment effect of interest in this context. I won't linger there or give you equations or anything like that, but I'll give you a high-level idea of what that is. You just heard, I guess, a lot about, or you probably already know a lot about the MGTX study. I'll very briefly talk about how we'll use that here. It's primarily for demonstration, not any sort of relitigation of that trial. I'll then go over 5 strategies for addressing rescue therapy. We'll talk about what is the strategy, and then I'll talk a little bit about how would you potentially apply this to some real data. And then after we've gone over the strategies, we'll look briefly at just some data analysis results to give you kind of an idea of how the results might differ depending on the strategy that you take. Then we'll conclude and talk about future work. Next slide. Yep. So yeah, I've got intermediate slides here. So the first part is, again, why should you care about this? So next slide. So from the causal inference perspective, there's really 2 ways you end up with biased treatment effects, 2 primary ways. So the first probably, it was a little more well-known. So this is confounding, or often talked about as baseline imbalances. So essentially, if you have some factor that affects whether you get the probability you get the treatment, and then also affects the outcome, and then thereby warps the observed effect. We address this by randomization, because that then, in most cases, or the simplest cases at least, your probability of getting the treatment has nothing to do with your patient or with your characteristics at all. So you end up with 2 comparable groups. The second way, though, that you can end up with biased treatment effect estimates is when things happen after randomization. We call these intercurrent events that may be related to the treatment assignment. And so at that point, randomization doesn't help you anymore necessarily. So essentially, rescue therapy is an intercurrent event. So the treatment that you receive, especially if it's if one is more effective than the other, is likely going to have some effect on whether you need rescue therapy, and then that may affect the endpoint, the ultimate endpoint. And this makes it difficult to tease apart the effects of rescue therapy from the experimental treatment. So some considerations are when rescue therapy occurs could matter. It might be that it's if you have, for example, it happens very early, then maybe those effects have worn off by the end of, say, a long follow-up. Whereas you might be even more concerned if you have more of a shorter follow-up, or those events are occurring closer to the time point where you've measured the endpoint. And so the key is the results can depend on both how you choose to handle rescue therapy and your definition of the treatment effect, and also the analysis you choose. I think next slide. So to give you some intuition, we could think about what would happen or what could happen under an effective treatment. So if you have a therapy that, or even if you have, say, 2 effective treatments and one is simply more effective than the other. In that case, you might expect that rescue therapy will be more likely to occur in the group has their MG less well controlled, so the control group. And that then you're taking these because you presumably you want to improve the patient's just general state and functional status. So that may then improve follow-up measurements. And so what you could see in that case is that the treatment and control groups actually look more similar than they should. And this is due in part to the rescue therapy, not because you say don't have an effective treatment. So in this example, you could end up with a biased treatment effect on average, that is a smaller effect than the true effect if you're thinking about a head-to-head comparison between two options. And then perhaps also another aspect of that would be decreased power, so you may miss effective treatments. There are other scenarios similar to this, but this is just one particular scenario that hopefully gives you some idea of what the problem is. Next slide. So we did in a paper that actually just recently came online that we published through this work. Part of what we looked at was looked at how studies were handling this in general in MG, and so we found various rates. We had as low as 1.5%, as high as 14.5%. Some of this may have been due to just differences in baseline disease severity. If you have a higher disease burden, you're likely to have more rescue therapy. And I think this may be especially important looking forward if we're wanting to, say, target therapies to individuals who don't have their symptoms well controlled with current therapies. And also from a regulatory perspective, you can't know what the rates are going to be prior to the study. And so you have to be, there's increasing pressure or guidance to plan for intercurrent events like rescue therapy well ahead of time. You don't want to be you don't want to get into the study, not have planned for it, and then have it arise, and that's not part of your protocol. Okay. Next slide. Okay, so the fun part, what are estimands? Okay, so this is how we're going to get rescue therapy into the endpoint definitions. Next slide. So I've given this as biostatistician for treatment effect in the population of interest. 4 out of these 5 components are things you're probably already thinking about. And you might be thinking about intercurrent events as well, but essentially an estimand is a very precise definition of your treatment effect. So it's not just taking a parameter from some statistical model. It encompasses your population. That actually, we saw some discussion of various, that myasthenia gravis is a heterogeneous disease. So you might be targeting therapies to specific subsets or subpopulations, and that matters. The endpoint isn't just, say, myasthenia gravis activities of daily living or some other functional endpoint. It's also encompasses when are you, what is the duration of the study? When are you defining this? Is this a change in baseline over 6 months or 3 years, as MGTX was 3 years, a little bit longer? And so that's also part of your endpoint. Treatment regimens, obviously, otherwise, what are you even doing with the trial? The population level summary, that's a little more of a statistical focus. It could be a difference in means. It could be a risk ratio or an odds ratio or something like that. And then the final one is what I think probably gets either ignored or just maybe not handled with as much precision as you might like, and that's the strategy for handling the intercurrent events. So that's primarily what we're talking about here. And so in general, what I would encourage you to do, and I guess regulatory bodies are encouraging, is defining your estimand first, including your strategy for intercurrent events, and then tailor your statistical methods to that estimand rather than trying to find a statistical method that you think might possibly help you deal with the intercurrent events. You want to be more targeted than that. Okay, and next slide. Yeah, so I'll just give a brief overview of how we're going to use MGTX. So it's for demonstration only. We're not relitigating that study or anything like that. Okay, next slide. So we're going to limit our analysis to the first 12 months. We'll look at MG-ADL instead of QMG. In that trial, if you're familiar with it, the statistical analysis that they used was quite complicated. We will avoid some of those details here. It's very interesting, but not relevant to this particular talk. So we're going to use it primarily just to demonstrate the application of some methodology and to look at how you would define these treatment effects. We'll define rescue therapy as plasma exchange or IVIG. And just to remind you, even the previous talk mentioned this as well, the comparators were thymectomy plus prednisone compared to prednisone alone. Okay, next slide. So based on our definition of rescue therapy, you can see that rescue therapy was about twice as high in the prednisone alone group, so 13 versus 7. Maybe against my better judgment, I put a p-value over here. I wouldn't be using p-values to decide whether you had a difference that's out of habit, but you can see, I guess, smaller sample size. And that's true actually in a lot of other cases when you're dealing with bias. Even if you were dealing, say, with confounding, you want to be careful about using p-values. You really want to base that on clinical expertise in terms of what you're controlling for. That's a separate talk. Give that another time. All right, next slide. Okay, so now what are the estimand strategies? How do we, what are we actually going to do with rescue therapy in these definitions? Okay, next slide. The most basic strategy is a treatment policy strategy. This is essentially ignoring rescue therapy, just not incorporated into the analysis at all. This is not invalid, but it's not usually the treatment effect you're looking for. And again, the reason is you can't separate the effects of the experimental therapy from that of rescue therapy. And part of the reason for this is that the implicit comparison, if you ignore rescue therapy, is that you're really comparing one group that is experimental therapy plus maybe rescue therapy as defined in protocol, and the other group is the control plus rescue therapy, maybe, that is if you need it. And so implicit to those comparators, rescue therapy is tied in there, and you can't really separate what's rescue therapy and what's the effect of the treatment of interest. So this is closely aligned with an intent to treat approach. So what is the average course of patients after randomization, perhaps? So if that's what you're asking, then this may be appropriate. But if it's not what you want to know, then this is probably not appropriate. Okay, next slide. So in terms of applying this strategy, again, we ignore rescue therapy. The endpoints, so if we're looking at, say, the MGTX trial, we could look at, say, change from baseline to 12 months in MG-ADL, or the final follow-up measure in MG-ADL. The statistical analyses you would use are ones you're probably familiar with, as regression for change in baseline, or ANOVA, or you may be familiar in two groups with a t-test. If you're looking at the final follow-up measurement, you're probably taking more of an you're adjusting for baseline MG-ADL as well, typically, is at least the recommendation in the literature. If you have a lot of missing data, you might use, or you're interested in the specific trajectories over time, you might use linear mixed models to deal with that. So pretty standard application, but none of it, rescue therapy is not entering into how you're designing the statistical analysis. All right, next slide. So hypothetical strategy is essentially asking what would happen if there's no rescue therapy. So this is more, I think this is one of the more common, and not just in myasthenia gravis, but elsewhere, approaches to take. And the reason for that is it really gives you, you're estimating the head-to-head comparison. So you're really trying to get at what would be the treatment effect if really the only difference between these two groups was one gets this experimental therapy, one gets this control therapy. And so it isolates the treatment effect from rescue therapy. A naive approach of this is often to just exclude patients who didn't receive rescue therapy. Those assumptions are typically untenable because they're assuming for that analysis to give you unbiased treatment effects. You're assuming that those who got rescue therapy were similar to those who didn't, and that's almost certainly not true. So more common is to exclude the follow-up measurements post-rescue therapy, and that will be true for another estimate strategy we'll see in a minute. And so it's basically saying, well, let's just use all the follow-up measurements that weren't quote unquote contaminated by rescue therapy. And so this imposes some missing data we then need to think about and deal with. All right, next slide. So how might we estimate this? So this is almost always either explicitly or implicitly kind of an imputation, or you can view it as imputation in some sense. The easy imputations are some like last observation carried forward is one. So take the last observation that you had before rescue therapy and pretend that's what you would have had at final follow-up. So that's a pretty strong assumption. Worst score imputation is similar, but instead of taking your last observation, we look at the population or the subset of the sample who did not receive rescue therapy and take the worst score, so say the least preferable change from baseline or the worst MG-ADL, and we give that score to people who had rescue therapy. So that's also a pretty strong assumption. So I wouldn't generally recommend either of those approaches. Linear mixed models or inverse probability of censoring weighting assume missing at random. I'm going to attempt not to get too much into the weeds of missingness here, but essentially you have three primary missingness mechanisms. One is that it's missing completely at random, which is informally that your missingness has nothing to do with the study. Missing at random is more of, well, it may have something to do with the study, but conditional on our observed data, the missingness would be not any longer have to do with the study. Basically, you're accounting for it by things you've measured. And missing not at random is if you don't have missing completely at random or missing at random. So it's basically, even if you have this, the missingness is due to things that you did not observe, which is kind of technical. So I would argue that this missing at random assumption is not probably reasonable for your for this setting, and there's actually some more recent papers in other contexts looking at this, and these models actually do they don't do as well in simulation studies where they're targeting the hypothetical estimand strategy. Another approach is a joint longitudinal and time to event model. I'll stay out of the weeds of that too if I can, but essentially what you're doing is still you have a linear mixed model to model the trajectories of MG-ADL over time, for example, but then you also model a time to rescue therapy model. And there's some disagreement about whether you officially call that MNAR or something else in the literature, but that's essentially, this is what it's doing. So I think this may be more of a reasonable approach for rescue therapy than the standard MAR models. All right, next slide. So while on treatment strategy is similar to hypothetical in that we typically exclude observations after rescue therapy, but the way that we define the treatment effect is this is what we would see prior to rescue therapy. Historically, this has been more relevant for death. So you might ask, well, how much improvement can you expect after 6 months or prior to death, whichever comes first? And the reason for that is it seems hypothetical strategy doesn't make a lot of sense for death, because you're saying if the patient had not died, what would their values have been? And that's just sort of, I don't know, that's all that relevant, and many people in the literature think this as well. So the key for myasthenia gravis and rescue therapy, we don't have the same issue with rescue therapy as we do with death, because you could potentially think about what would happen if you hadn't received rescue therapy. But you're basically saying the length of follow-up doesn't matter to you quite as much. So you're not tying your effect estimate to a landmark time point anymore. So you would be asking something like how much improvement after 6 months or prior to rescue therapy, whichever comes first. And so kind of confusingly, we can still use, like, for example, linear mixed models excluding post-rescue medication measurements. And I'm still sort of working on, I guess, the technical details to see how convinced I am of some of this. But my sense is that what the linear mixed model is actually estimating is probably while on treatment strategy due to the missingness assumptions discussed otherwise. You could use some similar models as last observation carried forward, for example, although there you're not really carrying it forward. So it's a different interpretation. There you're really interpreting it as, no, we're not pretending this is what they would have at the end of the study. This is what they have now before rescue therapy. And so that's the difference we're paying attention to. So it's a little more reasonable, although I probably being a statistician would still prefer something like a linear mixed model. But in any case, next slide. So composite strategies are where you build rescue therapy into the endpoint. And this is similar to what you might have seen. Many of you who've probably done trials may have used a composite endpoint. It probably was, I would guess, like several binary endpoints, and then you end up with a final binary endpoint. Here it's a little more complicated because we're not necessarily dealing with binary endpoints where we have some continuous outcomes and we have time to rescue therapy. So essentially, this is assuming rescue therapy is as indicative or similarly indicative of treatment success or failure as the functional outcome, for example, MGADL. And so the endpoint ultimately ends up being based on rankings. So for example, the worst outcome would be the first to receive rescue therapy, then the next worst is the second to receive rescue therapy. And you work through all those that had rescue therapy, and then you're ranking based on the functional measurements. So the best outcome then is going to be the individual with, say, the best change from baseline or the most optimal final follow-up measure for MGADL. Initial tests, or initially this was just a hypothesis test based on a Wilcoxon rank sum, which some of you may have learned about in your stats classes. The difficulty is there was no treatment effect, which I think in practice, we want a treatment effect. We don't just want to know that we had some small p-value. And the solution to that is, at least one of the solutions, is a win ratio. And so this is interpreted as pairwise comparisons between those in the treatment group with those in the control. So basically, you take every person in the treatment group and you compare them with every person in the control group. And for each of those, you look and see who had the better outcome. And so in the end, you end up with a ratio of wins to losses for the treatment group. So values above 1 would indicate that those in the treatment group essentially have a higher probability of having a better outcome than those in the control group. So it's no longer in terms of you don't have a treatment effect in terms of your functional measurement, but you do at least have a treatment effect that you can interpret in some way that's useful. Okay, next slide. So the last strategy is one that's more limited in practice. It's called principal stratum. There's two reasons primarily that I think it is limited in practice across disease areas. And one of them is just the methods are complicated and they have very strong assumptions. So it's kind of you don't necessarily want to work really hard to get a method programmed and working and then realize you don't believe the assumptions. The other issue is that it often changes the population of interest from what you cared about to some subpopulation. So you typically divide your patients, and you don't know this is the other issue. You don't know who is who with these subpopulations, but you're assuming there exists a subpopulation that wouldn't require rescue therapy no matter what treatment they got, a subpopulation that would require rescue therapy regardless of the treatment they got, a population that would require it if they were on active but not control, and a population that would require it if on control and not active. And so typically what happens is this first one is what gets estimated. So you're shifting from some population of myasthenia patients to just that subpopulation who would never require rescue therapy. And I would argue that's probably not what most of you are interested in. So we actually, when we won't look at this, I didn't program this for the other demonstration, but I wanted you to at least know that it exists on the off chance that you just disagree with me entirely and want to use it. Well, there you go. All right, next slide. Okay, so let's look at some comparisons for how the results differ when we applied them to MGTX. Again, this is just for demonstration. Next slide. So I've broken this into 4 sections. Again, we're excluding principal strata. So the first is the treatment policy. I've given hypothetical or while on treatment. I would argue most of these are probably more while on treatment given the assumptions than hypothetical. I've just strictly hypothetical. I've looked at the joint model, and we have the win ratio for a composite. So one of the things that you might notice looking at treatment policy is most of these are around -1.2. So that's saying by essentially 1.2 points on the MGADL scale, lower in the treatment group compared to the control group, so those getting thymectomy. And you'll notice that if you look at those treatment policy estimates, those are smaller than either the while on treatment approaches or the hypothetical approaches. So you can, and if you're thinking back to when we looked at the rates of rescue therapy in the 2 groups, you might recall that rescue therapy was in fact more common in those having prednisone alone. So we might not be surprised that if we then try to take account for rescue therapy, we end up with stronger treatment effects. I mean, in fact, one of the approaches in treatment policy didn't even get us to statistical significance. That's a separate talk about which methods are more efficient there's papers that exist on that, but so you could, if you had chosen linear mixed model with a random slope, if this was your study, have ended up with a non-significant result, and the reason might have been you were targeting the wrong estimand. You can see at least a little bit, there's depending on which method you're comparing it to, the hypothetical estimand was a little bit in between the treatment policy and while on treatment, although that doesn't follow exactly for comparing linear mixed model to joint model in this case. I would note also these first three, these are all treatment effects in terms of the functional measures. So you can interpret these as differences between groups in MG-ADL. So this change from baseline here in the hypothetical or while on treatment being right about 2 points on MG-ADL difference, essentially. Win ratio changes your interpretation. So this win ratio I've said changed from baseline. That's the functional measure we used for that analysis, but they're still ranking people based on rescue therapy as well. So it's still a worst outcome is the first to rescue therapy, best outcome is the best change from baseline. And so this is saying that you have that essentially those in the treatment group have about 1.9 times or about 1.9 times as likely to have the better outcome than the control group. And similarly for win ratio in the final MG-ADL. So this is nice. Win ratio kind of coming on the scene has been nice to get you another approach to dealing with intercurrent events that still gives you a treatment effect, gives you your confidence intervals, and doesn't just give you a p-value. So you can do something with this in practice a little easier. All right, next slide. All right, so what did we learn so far? So next slide. I'll skip over the review just because I think we're probably pressed for time. So I'll go to the next slide. So the conclusions, so the main thing, if you take nothing else out of this, I care less that you agree with me on every particular interpretation. What I really do care about is that you think about incorporating rescue therapy into the definition of the treatment effect. So just making sure that you're asking and answering the right question. So in this, being mindful of the interpretation of the statistical results. So the methods you're choosing ultimately align with specific estimand strategies. And so you want to make sure that the statistical analysis you're using is actually aligned with the estimand or the treatment effect that you've decided is what matters for the context that you find yourself in. And I think most of you are probably giving treatment effects in practice. That seems pretty common, but always put a plug in for don't just report statistical significance. You also want to look at what is the treatment effect, what is this interpretation, and also confidence intervals. Those give you additional information beyond what the p-value would give you about the precision of the treatment effect you've estimated. And so that's also going to be very useful for interpretation. Next slide. So future work. So where are we going from here? Next slide. So we're still working on some design for simulation studies to try to look at how well some of these methods perform compared to each other for given strategies. And also, as I mentioned, I'm still thinking about kind of formal definitions of some of these strategies and how those align with specific statistical methods. So we're going to look at those in settings that mimic MG trials because I think sometimes there's a risk that say, well, we did these simulation studies and we showed this method was best. Like, well, usually sometimes that's true. That's just informally better, but often how well a method works might depend on the context you're looking at it in. We're also thinking about the more I've dug into win ratio, the more I would like to work on that and seeing whether we can target it for other outcomes or intercurrent events. And to see the win ratio has the potential to really incorporate a lot of patient priorities, a lot of physician priorities that's been done in some other disease areas. And I think that would be potentially very useful in myasthenia gravis as well. All right, next slide. All right, yeah, I'm open for questions. I managed to somehow neglect my acknowledgement slide too. So I would just take a minute and thank my collaborators who we introduced at the beginning, and then also MGNet and the NIH for giving me the opportunity to do this work. Thank you.
That's great. So open to question. I think it's probably easiest if you could unmute and ask your question. So just to start off, I know this is not exactly, well, I'll just ask the question, you tell me. So in terms of influence of rescue therapy on primary outcome, so for MGTX, it was the area under the curve at 3 years over time, right? So the integration over of the prednisone dose and the QMG over a time period. So would that be less susceptible to these kind of rescue therapies versus a study that decides, for example, that the primary outcome measure is the status, the QMG of the patient at end of study?
That's a good question. I'm not sure. I think the main problem I might have had with trying to like wrap my mind around the primary analysis is how it would fit into the current strategies for dealing with rescue therapy. I think my intuition, I guess Gary Cutter is probably on the call, so he may have opinions about this too.
I am. I'll pipe in if you want me to.
My guess is that I would guess yes, but I wouldn't swear to that. Being a statistician, I'd want to like do some simulation studies. I guess because there, like you say, you have more. It might depend on when. I guess the answer is it would depend on when the rescue therapy happened and how long you think the effects of the rescue therapy are. I would guess that if rescue therapy effects were relatively short-lived, then having something like this over time would be more robust. But I'm happy to let Gary say what.
No, I think that's the right answer. I think, it depends, of course, on the kind of rescue therapy that you're doing. And, if you were doing a curative surgery for something, then it would have a bigger impact, especially if it occurred early. Keep in mind, by the way, in MGTX, we actually did do a penalty dose if rescue therapy was given on the prednisone. So we tried to take it into account over the short period that we felt like it might be impacting the assessment of the prednisone because something else was on board. But I think, as Justin said, if the rescue therapy is not too long-lived, then having it integrated over the entire period of 3 years, it'll have less impact. In the studies that are done today that are maybe, 26 weeks or less, the impact can be much, much greater.
Excellent. Anybody else?
Yeah, Henry, this is Richard Nowak here.
Yeah.
My hand is raised, but I'm not sure if that function is working. I'll just lower it.
Yeah. Oh no.
So
Go for it.
I think the question that you had, Henry, is whether or not there's a greater impact on the outcome measures, the clinical outcome measures like a QMG or an MGADL score as compared to another primary endpoint such as a reduction in steroid dose. So I'll just provide, I don't think it matters. I think it's more the duration of the study, like Dr. Cutter has just suggested. In our BEAT-MG study, it was intent to treat. We did not take into account, well, we did take into account, but it still was intent to treat, and we didn't consider folks a treatment failure if they received rescue therapy. However, in the placebo group, there was a approximately threefold higher rate of relapses requiring rescue therapy, IVIG or plasmapheresis, were the two rescue therapies allowed per protocol. And we found that participants were essentially equally able to reduce their steroid dose. So we did feel, I mean, it's hard to go back in time and change that particular trial design. We did feel that rescue therapy allowed for patients to be able to reduce their steroid dose. I mean, it's a little bit hand waving. And when we did our sensitivity analysis, we were just a hair short of declaring non-futility, taking into account that rescue therapy use. So I think that it can equally affect the steroid sparing or steroid reduction endpoint potentially, as well as an ADL or a QMG. And so I think, really carefully considering how we do analyze data, interpret data, where there are individuals that do get rescue therapy. So I really appreciate Dr. Leach, you bringing this forward and kind of thinking through, because as MG is a disease that has a varying course, we do expect rescue therapy. And then how do we interpret those clinical trials in that context? So that's my 2 cents. I don't think it matters if it's a steroid endpoint versus a clinical outcome measure.
Yeah, Henry, if I can just add, and thank you, Justin, for a fabulous talk. Richard, I can't recall if you didn't do sort of a separate analysis that adjusted or accounted for rescue therapy in some way. I think Justin's talk really highlights the importance of doing that, because it can dilute out the treatment effect, and we may be missing things. I'm thinking about some of the recent clinical trials where we sort of see statistical significance but not clinically meaningful effects, and there's very little discussion in most of these trial reports about how rescue therapy was handled, and I really feel this has been something we've been relatively blind to in the MG field. So great job, Justin.
Thank you. I'd also point out it wasn't the point of what Richard was saying, but I think it is important to point out he was talking about doing sensitivity analyses. And I think that's also an important thing. Probably many of you are doing that, but I wouldn't underestimate the importance of doing those. And still, I think it can be useful if you said, even if intent to treat is what you want to do, it can be useful to pick maybe one of these other estimand strategies that you think might be relevant, and plan for that sensitivity analysis. And that gives you an easier way to interpret the reason, like the meaning of the sensitivity analysis. Because I know it's sometimes hard to commit to a single analysis, but you have to. But yeah, anyway, just my plug for sensitivity analyses.
No, I completely agree. If I might just add one thing, based on you're bringing this forward and the research and further investigation to this area, how do you think that might change the current registrational trial design? So some of this is really artificial and directed or dictated by the FDA in terms of our MG-ADL change from baseline as the endpoint. And I think there are folks that think that's a good one, and some of us out there think that that's probably not the best endpoint for investigating the effectiveness or efficacy of medications. Do you have any thoughts on that? I know it's a little bit open-ended and not a specific question, but is there something that we should be doing to convince or to bring the regulators along in terms of the endpoints and outcomes that are used in clinical trials?
Yeah, that's a good question. I think in terms of these estimand strategies, those are popping up from regulators as well. So I would anticipate that in future, it will probably be more likely that you need to incorporate, like, to say how, and not just with rescue therapy, but with intercurrent events more generally. I'm not sure I have a good answer for you on convincing them that you should use something other than MG-ADL. Although, I mean, potentially, and there's pluses and minuses to things like a win ratio, but a win ratio can also incorporate. It doesn't necessarily force you to choose a one outcome. It lets you build in and say there's more that we care about in terms of what patients care about, or what we care about as or what you would care about as physicians. So I think in the long run that you may be able to make the case for something like a win ratio as well, if you can make the case that it actually is taking into account what patients actually care about. And
there are two concerns though with the win ratio, of course. One is, what does it mean? It's really just a test of the difference between the two groups. And B, more importantly, what the regulators are worried about is components going in different directions.
Sure. Yeah, you would probably have to, well, I mean, I do think the only pushback I would have there is I do think giving a ratio, like, yes, it's harder to interpret, but it's better than you at least get some measure, as opposed to just a p-value. But yes, that can be, that you might have those going in opposite directions is something you would probably have to build into at least some sensitivity analysis. I would definitely agree with that. That can be a problem.
Okay,
I believe
Can I
Rhonda actually
just
has a
add one last
question.
thing?
okay,
Henry,
real
please.
quick.
Oh, real quick. So is there anything that we in MGNet can do to aid by providing any data sets, et cetera? Maybe we can take that offline. So sorry, I know I'm gonna derail the schedule, but maybe we can take that offline.
Sure, I'm happy to discuss anything and everything offline.
Thank you.
I
All
think
right,
two people have their hands up, Rhonda and Taylor.
Hi.
Oh,
Oh, sorry, I didn't mean to. Is it okay if I go? This
Yeah.
is Rhonda. Okay,
go right
thank
ahead.
you. Thanks. This is Rhonda Sesniak from the DMCC, and I just wanted to say thanks, Justin, for a nice talk. And the question I had was kind of related to just what was asked previously, but I'm wondering if you've given some thought in your future work with the joint models to looking at multivariate outcomes. So kind of mixed outcomes, and if that could help with your win ratio, just in terms of how you prioritize or look at the influence of certain outcomes. So if you didn't want to stick, for example, with something strictly like MG-ADL, if you could put in other outcomes. And I'm just kind of thinking that could be potentially maybe like a twofold strategy that could expand, for multiple diseases. So I don't know how much of that you've thought about. I know that's your future work, but if you just have a couple thoughts on that.
Right. I guess yes and no. I've thought about using joint models for a joint treatment effect because I have seen that in the literature. I haven't thought as much about extending it into like a multivariate case. But I think that's an interesting approach. I know you do a lot of work in that area. So I think I'm still, in some ways, I'm still wrapping my mind around the joint model. It's fascinating, but complicated.
I think I'll just say really quickly, I think it's kind of interesting because I know with the win ratio, sometimes there's difficulties in prioritizing events. And it seems like if you could kind of get at it, in a statistical way to look at different outcomes or events, simultaneously, that might be interesting. But thank you.
Yeah, thank you.
I think Taylor Bauman also had a question.
Okay, go for it. We'll just extend this into the break a little bit, but we'll make sure everybody gets 30 minutes. So, Taylor?
She might have put her hand back down.
Yeah, okay. Sounds good. She can definitely email Dr. Leach with questions. So that's
I guess I
fantastic.
could drop I don't think I have my email on there, but I can drop it.
Oh, yeah, that would be great. Thank you. And so let's move on to our next speaker, and Dr. Massey. I'll just tell you, you will have a full 30 minutes. So if we could, right. There we go. Excellent.
Thank you. Thank you, Dr. Kaminsky, and good morning to everybody, and good afternoon to people from Europe. My name is Gianvito Massi, and I'm an associate research scientist and neurologist in Dr. Kevin O'Connor's lab at Yale. And I'm glad today to speak about our recent work on mask-specific IgA, a novel autoantibody isotype in myasthenia gravis. A little note before I start my presentation, you're going to see several animations in the first part of my presentation. So thanks for your patience, and thanks, Anosha, for advancing the slides for me.
First.
So next slide. These are my disclosures. Next slide, please. So we all know that MuSK is the autoantibody target in a subset of patients with myasthenia. And I will start my presentation with a deep dive into the immune mechanisms underpinning MuSK myasthenia gravis pathology. So MuSK is a transmembrane receptor expressed on the muscle end plate that consists of several extracellular domains, 3 immunoglobulin-like domains, 1 Frizzle domain, and 1 intracellular kinase domain. Next slide, please. So MuSK interacts with 2 proteins, LRP4, its co-receptor, and agrin, which is released by motor neurons. And the 3 proteins form a complex. Next image, please. And this structure, which consists of the MuSK, LRP4, and agrin complex, has been resolved recently using cryo-EM. What we know is that LRP4 acts as a clamp that will increase the binding affinity between MuSK and agrin. Next image. And this structure will assemble into higher order oligomers, which will promote MuSK dimerization, phosphorylation of the kinase domain, and a complex signaling cascade. So what you can see here on the right are acetylcholine receptor clusters. Next image. But when MuSK is activated, AChR clusters are formed on the muscle end plate. And we can experimentally visualize this signaling cascade in vitro. Next image. When we incubate myotubes with agrin, the agrin LRP4 MuSK complex signaling is activated. And what you can see here are bright areas representing AChR clusters on muscle cells. Next. So these clusters are effective for neuromuscular transmission. However, in MuSK myasthenia gravis pathology, MuSK antibodies can affect cluster formation. Next slide. So what we know is that MuSK IgG4 antibodies are produced by MuSK-specific plasmablast. And because of the IgG4 isotype, these antibodies are quite unstable. Next. MuSK-specific IgG4 can engage with other unrelated IgG4, exchanging half molecules. So in the next image, what you're going to observe is that the final antibody is a bispecific and functionally monovalent. That means that the majority of MuSK-specific IgG4 autoantibodies will have only one arm that will be specific for MuSK. Now, if we think about the pathology of MuSK myasthenia, next image. Next. MuSK antibodies will bind to MuSK and they will interfere with LRP4 interaction. This will eventually lead to will prevent activation of MuSK. Next. And AChR clusters won't be formed on the muscle end plate. Next image. So what we can experimentally visualize here instead are myotubes that are incubated with agrin and the MuSK-specific Fab, which will mimic the IgG4 functional monovalency. And as you can see here, compared to the first image, you don't see cluster formation. So this is the main mechanism through which MuSK IgG4 autoantibodies mediate pathology. Next slide, please. So beyond IgG, IgA antibodies represent the second most abundant immunoglobulin in serum. And we know that IgA antibodies play a pivotal role in mucosal immunity. Next. However, there's also increasing evidence from other autoimmune conditions suggesting that IgA antibodies can also contribute to neurological and non-neurological autoimmune mechanism. Next, please. And here I just highlighted some examples coming from studies on multiple sclerosis, MOGAD, and autoimmune encephalitis, in which the role of IgA antibodies have been investigated. Next slide. So my research questions were 2. Number 1, we were interested to investigate whether in an IgG4-driven disease like MuSK myasthenia, MuSK-specific IgA antibodies could be present in circulation. And number 2, if present, we also were interested in exploring the role of these antibodies in the disease. Next image. So in order to answer these questions, we tested 26 patients with MuSK myasthenia with available longitudinal samples and controls. And we performed a MuSK cell-based assay in which MuSK is expressed on the cell surface and antibodies from patients are added in this experiment, and an anti-IgA secondary antibody is then incubated with cells to detect the presence of MuSK-specific IgA. Next. As screening methods, we use both flow cytometry and microscopy. Next slide. So let's dive into the results of our experiments. We found 8 samples from MuSK-MG patients who were positive for MuSK-specific IgA, corresponding to 3 out of 26 patients that we tested. Next image. All samples with MuSK-IgA antibodies were also positive for MuSK-IgG antibodies. However, we did not find a correlation between IgG binding and IgA binding. Next slide. And here I wanted to show you some representative image from fluorescence microscopy. On the left, you can see cells that fluoresce green because they express MuSK-GFP, but you don't see any signal coming from the IgG and IgA channel, because in this case, you're looking at a control. This is healthy donor. Next image. Here instead, you can see a patient with MuSK myasthenia gravis harboring antibodies of the IgG class that you can see in light blue. Next image. In this case, instead, I'd like to show you a patient in which antibodies of the IgG and IgA class are present. And at the bottom, you can see a very nice colocalization, for example, between the red signal, which represents IgA antibodies, and the green signal, which represents expression of MuSK on the cell surface. Next slide. So of the 3 patients with IgA antibodies, in one, longitudinal samples were collected spanning 12 years of disease activity. And so I wanted to select this case because I think it's an intriguing case study. What you see here is the longitudinal tracking of MuSK-specific IgG antibodies in blue and IgA antibodies in red in a single patient. And the orange dashed lines represent rituximab administration. So both IgG and IgA level decreased following CD20 depletion. Next image. Next. But if you look at the relative reduction of IgG and IgA levels in the same patients on the right, when the graphs show the superimposed images, you can appreciate that the reduction of IgG following CD20 depletion is greater compared to the reduction of IgA, suggesting that MuSK-specific IgG and IgA B cells may have different susceptibility to CD20 depletion. Next slide. And so in order to study the molecular characteristics of these antibodies in details, we leveraged and established a B-cell culturing approach in our lab. I won't go over all the details here, but what I want to highlight is that we were able to isolate B cells from a MuSK patient and to clone recombinant monoclonal autoantibodies. So basically, from single B cells, we were able to generate patient-derived recombinant monoclonal autoantibodies that target MuSK and are of the IgA class. Next slide. So from the 3 patients, we generated 1 monoclonal autoantibody per patient. The subclass of for all these monoclonal antibodies is IgA1. What I wanted to highlight here, if you look at the column in which you see numbers, those numbers represent the amino acid replacement in the variable region of the heavy and light chain of each antibody. And so these numbers basically mean that these antibodies are heavily somatically hypermutated and derived from antigen-experienced B cells undergoing affinity maturation. Next slide. So we wanted to test the molecular functional characteristics of these antibodies. So on the left, you see the 3 antibodies that we derived from the 3 patients in orange, green, and blue. And we also used a control in our experiment, MAB01A, which is an SHR-specific MAB that we subcloned into an IgA1 backbone. Next image. So first of all, we were interested in assessing the binding to full-length MuSK of this antibody. And as you can see here, all 3 antibodies could bind to MuSK. And this is something that we confirmed both using flow cytometry and microscopy. Next image. And here you can observe the binding curves of these antibodies over a wide range of dilutions. The red line corresponds to a positive control, which has sub-nanomolar affinity for MuSK. And as you can observe, all the 3 antibodies, the orange, blue, and green one, have similar relative binding strength. However, in the further experiments, we also observed that these antibodies were strikingly different. Next image. As I said before, MuSK consists of 3 immunoglobulin-like domains and 1 Frizzle domain. I'm talking about the ecto domain of MuSK. And we performed an epitope mapping experiment, which demonstrated that the 3 antibodies bind to 3 different domains of MuSK. And because IgG and IgA coexist in sera, then we also were interested in assessing whether the MuSK IgA antibodies could compete with MuSK IgG4 antibodies for binding. And on the right, you can see a heat map that summarizes the results of a competitive binding experiment. When you see the black squares, those correspond to condition in which an IgA mAb can compete for an IgG4 mAb for binding, indicating that MuSK-specific IgA and IgG4 autoantibodies can bind to the same or partially overlapping epitopes. Next slide. Next, we were also interested in looking more deeply at the B cells. Now, we know that each B cell is quite unique because the variable region of each B cell receptor derives from a unique DNA sequence. So the variable region can be compared to a fingerprint. And B cell receptor sequencing is a powerful tool in order to search for clonally related B cells. Next image. Which basically represent B cells that share a common ancestor with a B cell of interest. Next image. So using BCR sequencing, we were able to identify a large family of B cells. So what you can see here is our mAb, MU1 IgA, that we generated from a patient. But we also found that this mAb is actually part of a bigger family, including 24 clonally related B cells. Now, these B cells were found in time points spanning over 6 years of disease. And during these 6 years, rituximab was administered 3 times, further corroborating the hypothesis that MuSK-specific IgA B cells can be quite resistant to CD20 B cell depletion. Next slide. And finally, we were also interested in assessing the functional effect of these IgA autoantibodies on cluster formation. As I said before, because of Fab-arm exchange, IgG4 antibodies will bind to MuSK with a single arm. Next image. However, IgA autoantibodies are bivalent antibodies and have the potential to force dimerize MuSK, inducing phosphorylation of the kinase domain and clustering of the acetylcholine receptors. Next image. So in this experiment, in fact, we demonstrated that 2 out of 3 IgA autoantibodies, the orange one and the blue one, were able to phosphorylate MuSK, similar to agrin. And this is something that we were able to demonstrate using immunoblot. Next slide. We also incubated MuSK-specific IgA autoantibodies with myotubes, which also showed that those 2 antibodies, again, the orange and the blue one, were able to promote AChR clustering compared to a control mAb, 818C5, which is a MOG-specific antibody, suggesting that these antibodies can behave as MuSK agonists, activating the MuSK signaling cascade. Next slide. However, when incubated with agrin, the same 2 antibodies were able to reduce agrin-dependent cluster formation, indicating that these antibodies can actually show pathogenic potential in vitro. Next slide. And so in the last slides, I will show you how the characteristics of these antibodies are strikingly different and depend on the experimental context that we use. In this experiment, for example, we decided to incubate myotubes with the MuSK IgG4-derived Fab and agrin to mimic the MuSK IgG4-driven pathology in patients. And then we added an individual MuSK IgA monoclonal autoantibody to assess the interference of this antibody to the system. Next image. Now I'll guide you through the results of this experiment. What you see here, indicated by a purple or violet arrow, is the number of clusters of acetylcholine receptors when myotubes are incubated with a MuSK-specific Fab. Because the Fab is highly pathogenic, basically we don't see clusters at all. Next image. However, when we add individual IgA autoantibodies, these antibodies can partially rescue cluster formation. So not only do these antibodies behave as mask agonists, but they can actually antagonize mask IgG4 pathogenic monovalency in vitro. Next image. However, then we went back to the serum samples and we performed an epitope mapping experiment using serum. Here on the left, you see the different mask domains, while on the horizontal line, you see samples. Next image. And we were able to demonstrate that in some patients, IgA autoantibodies targeting mask could bind to different domains, suggesting that the circulating mask-specific IgA autoantibody repertoire shows evidence of polyclonality. Next image. Because of these results, then we decided to incubate mask IgA autoantibodies together. So in this experiment, you can appreciate how individual IgA autoantibodies behave as mask agonists and can promote SHR clustering. The SHR clusters are represented by those bright areas that you can see on these microscope images. However, the combination of two mask IgA autoantibodies actually resulted in inhibition of cluster formation. Next image. In the presence of agrin, individual IgA autoantibodies could partially reduce cluster formation, but again, the co-incubation of two antibodies significantly diminished cluster formation, indicating that the combination, the interaction of multiple antibodies could actually be high pathogenic. Next slide. So to summarize, we identified mask-specific IgA, a novel autoantibody isotype in mask myasthenia. We detected IgA autoantibodies in 12% of patients in our cohort. And we also have intriguing data indicating mask-specific IgA B cells can also be quite resistant to CD20 B cell depletion. Next. At the monoclonal level, the IgA monoclonal autoantibodies exhibited different binding to mask. And from a mechanistic standpoint, some clones behaved as mask agonists, promoting SHR clustering and antagonizing mask IgG4 pathogenic monovalency in vitro. Next, please. However, we also found that the combination of two IgA clones resulted in potent clustering inhibition, indicating a potential cooperative mechanism through which bivalent clones may enhance pathology. Next, please. So our study on mask IgA antibodies obviously raises new questions. In mask myasthenia, but also in other diseases, the role and the origin of IgG4 autoimmunity are still undetermined. So because IgA B cells can theoretically class switch to IgG4, one question is whether mask-specific IgA B cells can represent a cellular reservoir for mask IgG4 autoimmunity. And because we identified IgA autoantibodies and IgA antibodies are mainly found on mucosal surfaces, the next question is whether mucosal triggers can fuel an autoimmune response against mask. And finally, having shown that IgA autoantibodies can also have pathogenic potential in vitro, one can wonder what is the efficacy of IgG4 targeted treatment in patients with IgA autoantibodies. Next, please. Finally, I'd like to thank Dr. Kevin O'Connor for his continuous support, Dr. Richard Nowak for the samples, and my colleagues and all of those who contributed to this study. And finally, I'd like to thank our funders and thank you, all of you.
Okay. Wow. You blew my mind with that. Lots of questions, but I'll open up to everybody else on the line here. So feel free to unmute. Dr. Sullivan.
Yeah.
Go ahead, Betty.
Okay. Very interesting. Very interesting. So back to the question of the 11% patients with these IgA antibodies, the response, were they better, worse? Did how do you correlate back to the clinical response to rituximab?
Mm-hmm. Yeah, this is something we're actually analyzing right now. I have to say though that those patients first of all, receive like different treatments. So what I showed you, like the intriguing case study that I showed you is from a patient that was treated with rituximab over time, but in the other two patients that were positive for mass-specific IgA02 antibodies, rituximab was not given. So we don't think that rituximab is the reason why we see a rise in the mass-specific IgA02 antibodies. We are also interested in assessing whether there's a correlation between disease severity and the presence of mass-specific IgA. Given the limited number of patients, obviously, we don't have like enough statistical power to assess this correlation, and we're actually collecting more samples. However, we have observed a trend suggesting that masked IgA02 antibodies can actually contribute to disease severity.
Thank you.
Anyone else? Yeah, Vern? It's all yours, Dr. Jewell. You can unmute. Yep.
Sorry, I got to unmute.
All right.
Is there an association between disease duration and MuSK IgA?
Yeah, that's interesting. So based on the data that we have right now, I cannot give you a final answer. Also because in one case, the patient was lost at follow-up. So we basically don't know whether like the patient achieved stable clinical remission or not. But we have data based on the 3 patients that we have right now suggesting that actually the IgA antibodies can contribute to like a longer disease duration, at least based on the patient that I showed you that is currently seen like in the clinic, and this patient still receives CD20 depletion because of continuous relapses.
Thanks.
So Dr. Sikorsky?
Okay, great. Hi, great talk. I just have a quick question about if you've tried to phenotype these B cells that are IgA positive, and what do you think they are?
Yeah, great question. This is something we're interested in. We haven't phenotyped those B cells, but we have some clues that can basically at least help us formulate some hypotheses. So as I showed you, the masked IgA levels diminished over time, but compared to the IgG levels, the IgA antibodies were quite stable. And so basically that could mean either that the masked IgA02 antibodies are mainly produced by plasma cells that are CD20 negative. But we also know that that IgA B cells mainly reside in the Paris patches in the gut. So another like hypothesis is that those B cells can also be produced by plasmablast, but they are less affected by CD20 B cell depletion because they reside in specific tissues. And we know how like compartmentalizations and the tissues can affect the response to like biologics like CD20 depletion. So again, this is something that we are still investigating, but right now we're like open to both hypotheses.
Okay, I see Dr. Kushner.
Yes, I'm just interested in this development of these B cells within the gut. I mean, is there a thought that they're not targeting the neuromuscular junction, but these antibodies are all, primarily targeting something in the gut? Any look at that? Did you, potentially looking at mice would be very interesting to start, analyzing in their gut for these, lighting up with antibodies.
Mm-hmm. Yeah, we are like quite interested in that. Also because, again, we don't know much about why IgG4 antibodies develop over time. There are like obviously some clues in some cases like chronic vaccinations or like allergens. But there's also like an interesting relationship between the development of IgG4 antibodies and oral tolerance. So suggesting that maybe something in the gut is triggering the production of IgA and perhaps IgG4 autoantibodies. We are interested in this, and we are actually looking at whether the IgA monoclonal autoantibodies that we generated can bind to gut bacteria. This is something that potentially could open new pathways and will improve the understanding of the disease. And again, also, I think given the fact that we were able to isolate those monoclonals, now these MABs represent wonderful tools to investigate specific mechanism of the disease. And again, this is something that yes, we're really interested in looking more deeply. Thanks.
Just to follow that up, with the patients with MuSK, is there any symptomatology that would suggest there is an autoreactive process occurring in the gut?
No, at least we don't have any data indicating that something is happening at the gut. However, there are also other diseases in which the role of IgA antibodies have been explored and is I would say more clear, for example, in lupus or rheumatoid arthritis. And there's not always a correlation between the production of IgA B cells and then the inflammation of the joints, for example. So we don't have, yes, maybe it's also possible that something is triggered in the gut, but there's no real inflammation of the mucosa, and something is related more to external or environmental triggers. Again, this is something that is really interesting, and we will investigate in the future.
Excellent. So actually, maybe you could just speculate on this. So you've described the IgA having clustering signals, the IgG4 unclusters, but then you have the agrin. So I don't know if these also potentially cooperate in some way as well. I mean, the complexity of how AChR antibodies injure the postsynaptic membrane is obviously more complex than what he thought so 5 years ago. So, you know, in some situations when the patient is doing well, these IgAs are clustering, and sometimes when they're not doing well, they're unclustering. Is it the ratio of all these?
Yeah, that's a good question. So obviously there are a lot of variables to consider. For example, what I showed you is that 3 patients had MuSK-specific IgA antibodies, but when you look at the specific characteristics of these antibodies, the 3 antibodies bind to 3 different domains of MuSK. And we know that the specific domain is is crucial because, for example, there are a couple of wonderful and outstanding articles that have been published recently on the potentially therapeutic role of MuSK-specific antibodies that target the Frizzle domain. So we know that the Frizzle does not interact with the MuSK-LRP4 pathway. So by binding to the Frizzle domain, the antibodies can activate MuSK, promoting SHR cluster formation. However, in our case, we have antibodies, one antibody bound to the Frizzle domain, the other 2 antibodies bound to the IgG1 and the IgG2 domain. So we know, based on our study in the in vitro experiments, that yes, these antibodies are able to induce MuSK phosphorylation and cluster formation. However, when we incubated these antibodies with agrin, we also saw a reduction of agrin-dependent clustering. So it seems that these antibodies can behave as MuSK agonists, but at the same time can also interfere with agrin formation. So overall, I would say that we have some data suggesting that these antibodies can be pathogenic, and they can also share similarities with the IgG1, 2, and 3 antibodies that are present in some patients with MuSK, for which case there are, again, some pieces of evidence from the Oxford group indicating that bivalent antibodies can also be pathogenic.
Excellent. Thank you so much. So we'll move on to our next speaker. Okay, Dr. Smith, you are up.
Thanks, everyone. I'm very excited to speak with you today, so I really appreciate the opportunity. We'll be telling you about our pilot project defining immunogenic signatures of AChR-mediated myasthenia gravis. Next slide, please. So the philosophy of our lab in general and sort of how we got to myasthenia gravis was this idea that response to things are variable and antibody responses and the foundations of them are not really understood. So I usually exemplify this using the example of vaccination. So we all know that responses to vaccination can vary widely depending on the antigen and the formulation. Understanding the factors that influence this response is extremely limited. And without understanding maybe the host contribution to that response, the potential of a universal vaccine or universal treatment for anything antibody related may be problematic. Next, I have quite a few animations here. So our best example for this right now is MMR, so the measles, mumps, and rubella vaccine. 90% of children receive this vaccine every year. That's over 3 million a year. Up to 10% won't respond. About 300,000 children lose their mom. That's a bit of a terrifying number, particularly when you bring in the unvaccinated populations as well. But only 20% of that response variation is currently understood. Next. So our lab has developed several assays that I'm going to tell you about today to provide new evidence that suggests that the actual genetic background, the foundation of the genes that we encode in our immune response influences immune response potency and potentially pathogenic antibody formation. Next, please. So as has been well described already by many of the talks, you can go ahead and hit the next animation. Antibodies have a myriad and varied effector functions. And what I really want to hone in on today is actually the constant domain. So we know that the variable domain, which we profile routinely in adaptive immune receptor repertoire sequencing, like was just so nicely presented by the O'Connor lab, binds the antigen. It's the business end of the molecule. But the tail of the antibody, that constant region domain, is really responsible for a lot of those downstream effector functions, including antibody-dependent cell cytotoxicity, phagocytosis, and activation of complement, in particular for today's talk. Next, please. So, and you can go ahead and click through. Thank you. On the left is a simplified schematic of what the antibody loci looks like in our genome. But on the right is an actual schematic of an antibody locus and the complexity of what is actually encoded on chromosome 14, particularly in the heavy chain of the antibody. So those green boxes represent those variable domains, so the V in the VDG phenotype. The red actually represent SIFO genes, which used to be variable genes that were somehow rendered ineffective through evolution or segmental duplication. Diversity genes in navy, and down there on the second to bottom line, there's a small yellow box with the 6 J genes that we know that make up that recombined VDJ heavy or variable region on the heavy chain. Now highlighted in blue at the very bottom, the baby blue, are the constant region encoding alleles, and that was referred to again in the talk that just came before, which is an excellent introduction. These genes encode the IgG 1, 2, 3, 4 subisotypes, A, E, D, and M isotypes. Next. There has been a lack of association of disease susceptibility or resistance associated with the plethora of variation that happens at this locus, and this is mainly due to the era of GWAS that uses immunoarrays. You can click through the next 3. A lot of immune loci, immunogenomic loci such as HLA and here, have relatively good representation of population variation in these arrays. So HLA has 9,300 SNPs that are represented by the oligos in these arrays. Even the NK cell receptors up here have hundreds of variants represented, but IgH only has 5. So for a locus that is over 1 million base pairs long, that we know has extensive variation, it's very poorly represented in many of the assays that are used to determine genetic determinants of disease resistance or susceptibility. Next slide, please. So together with my colleague Corey Watson here at University of Louisville, you can click the next one, please. We've developed 2 assays, that we're calling iGenotyper and FLAIR-seq to really look at population level variation at both the DNA level in the germline and how that translates to what's circulating in the repertoire. So this first tool, which I'll describe in just a minute, is iGenotyper, and really our model is that different populations encode very dramatically different antibody genes or alleles at the basic foundational DNA level that then translates into differential repertoire composition, which then leads to differential response or susceptibility to disease. All right, now you can go to the next slide. This is a very complicated figure. I don't really want to belabor the point, but what I do want to talk about is how this shows the variation of the germline locus. So many of us in our Immunology 101 class are taught that we all have the same VD and J genes. We just recombine them differently through a random stochastic process, and that's why we have differential responsiveness. Our data, which is now numbering in the thousands, but what's represented here is 154 individuals, shows that that is definitely inaccurate. And the way I can mostly highlight this, so if you look at this figure, there are the genes, which are at the top in different colors. These are the VD and J genes that encode the variable region of your heavy chain. Below that are the structural variants, and these are represented by those red dots. Those represent deletions that were identified in at least 5 individuals out of 154 in this cohort, in this paper. And then the sort of upside down triangles, those represent insertions of new genes represented in at least 5 individuals. What you can see most importantly is that not all individuals are the same. In fact, some people encode these massive deletions that will move up to a third of the variable regions that are available for incorporation into these genotypes. Next, please. Not only are genes present or absent between individuals, but then each of these genes comes with sort of an allelic flavor to them, right? Allelic variation and copy number variation as well. And that's represented in this bottom graph, where you can see that certain of the V genes we identified over 10 novel alleles in these 154 individuals. What this really drives home is that we don't understand the full breadth of variation at this locus. It's been completely underestimated in our models of immunity thus far, and that a greater understanding of this could identify potential allelic variants or genes themselves that are necessary for proper immune responses or maybe modulating pathogenesis. Next slide. In addition to the germline focus, which is done with an oligo-based capture assay, very similar to an exome, but targeted towards the IG locus, we've also developed an assay we're calling FLAIR-seq that targets this transcript. So again, the previous talk talked about antibody repertoire sequencing really well. A lot of the current methods currently use these primers that are identified in the green box or constant region 1 exon, especially the one sort of to the left that captures the isotype signature that can tell you whether it's an IgM, IgD, IgA, or IgE. But sub-isotype signatures are a bit downstream into the constant region. And of course, those motifs that may determine functional differences would be further down in that CH1, CH2, CH3 constant region exon encoding region. So current technologies can give you isotypes. They cannot identify sub-isotypes, and they certainly can't separate those sub-isotypes into alleles. And that was our focus in developing this assay. Next slide. If you can click through all the way, that would be great. So yeah, so for us here, I'm representing just a few of the common assays that are used for AIR-seq. So adaptive biotechnologies, right, really focuses on that antigen hypervariable, that CDR3 region that interacts with the antigen. It's a very small amplicon. There are primer-based methods that use primer pools to amplify the entire variable chain. And then these rapid amplification of DNA ends, such as VDJ race from Takara or NAD, that's used to grab that partial CH1 to gather isotope information. You can click once more. So our method, FLAIR-seq, actually, one more time. Thank you. It uses long-range sequencing to actually scoot that primer back. So we start out with a very similar race method, but then we amplify the entirety of the transcript or near entirety of the transcript using a bar-coded primer in CH3 that allows us to resolve the entire antibody transcript as a single molecule. We sequence this data on, this is a Sequel II instrument from PacBio, but now we have the Revio system from the PacBio. The way that PacBio sequencing works actually uses multiple passes over the same molecule to enhance the overall accuracy. Because these are relatively short molecules, the polymerase 1.5 to 2 kb, the polymerase goes around these molecules more than 100 times, creating an incredibly accurate circular consensus read for us to use for downstream analysis. In fact, many of the reads that I'll show you today have a Q value or quality value of over Q60, which is equivalent to 99.999% accurate. So 3 orders of magnitude more accurate than you get from other sequencing technologies. All right, next slide. So this is what the data actually look like, and I don't want to belabor the point, but this is an alignment view of an IgG4 molecule in the integrated genomics viewer. The variable region, very variable. You can see it on the right-hand side represented by this colored stack. And then the constant region is represented by the exons in gray here on our screen. And what we do see is even though the constant region is less variable than the variable region, there are still allele-defining SNPs in these exons that allow us to segregate and be able to look at allele-specific variable region coverage when everything is linked together with a single molecule. Next slide. And in fact, when we validated this approach on 10 healthy individuals and looked at constant region usage and constant allele usage across these 10 individuals, so these are not myasthenia gravis individuals, just healthy PBMC that we bought from Stem Cell Technologies. On the left-hand side are the sample numbers, and across the top are the different sub-isotype alleles we were looking at for IgG and IgM. What I want to highlight is that any box that you see has a dot was seen in both the DNA and RNA data, so the iGenotyper I talked about initially and the FLAIR-seq data, but any allele that you can see labeled with a color that includes an FL in the name, those are all novel. So in 10 individuals, 87% of the alleles that we found were novel compared to the IMGP database. We also saw a number of duplications, so genetic structures that had duplicated in the constant region, leading to more than 2 alleles expressed, right? So usually we have a maternal and a paternal allele. In the case of IgG4 in particular, we have found in our population-based sequencing that about 30% of individuals contain at least one duplication of IgG4, leading to at least 3, if not 4, separate IgG4 alleles circulating in these individuals. Next slide. So the motivation for this project that I'm talking about today, right, is that we know that AChR-MG is mainly mediated by IgG1 and IgG3 autoantibodies, and that there are 3 main mechanisms driving this disease. So if we could go animate. So as has already been discussed in the first talk, right, a lot of this is focused on complement-mediated degradation, internalization, and functional block. Those are the known mechanisms of pathogenesis. I mean, you can click twice more. Specifically for us, very interested that one of these mechanisms is FC-mediated. Ours technology of doing antibody repertoire profiling using FLAIR-seq would be the only technology that could associate the G1 or G3 specificity with a particular clonotype to identify and weed out possible pathogenic antibodies. The translational potential that we see here is that if we can identify genomic variants associated with, for example, complement-mediated pathogenesis, that would help us pair the appropriate treatment regimen with someone that would benefit from it, right? So if you have AChR-MG, but you're using one of these other mechanisms of action, a complement-mediated inhibitor isn't going to necessarily help you, but may have more detrimental effects. So if we could use some of these genetic signatures to parse patients according to their mechanism of action, or identify potential biomarkers of risk that are associated with autoantibody development, then perhaps we could help bin you differently clinically in the next slide, please. You can click through this. So we used 48 healthy individuals and 48 myasthenia gravis samples from the biobank accessed through our collaborator, Dr. Kevin O'Connor at Yale. We performed both the DNA sequencing, see here on the left, to get ground truth personalized reference IG haplotype. And then we did the RNA sampling or the repertoire profiling to see how the variants that we see in the genotype are reflected in the antibodies that are actually expressed in the repertoire. Next slide. The first thing we wanted to do was to see what kinds of antibody signatures we saw. So the first thing we did is to look at the diversity index. This is a statistical model to look at the degree of clonality in your sample. So are there B cells that are being stimulated? Are they driving clonal expansion, suggesting that they are responding? So you can see that between the healthy donor and MG individuals, we have statistically different clonal expansion, far less diversity in the MG individuals and those healthy donors, suggesting clonal expansion. Next slide. Using the FLAIR-seq data, we can then parse down where that clonal expansion is happening, in which compartment. Is it IgG1, 2, 3, or 4? And what we see is that there's more significant clonal expansion in G1, G2, and G4, specifically in G4, which is very interesting to us, suggesting that there's something happening in those specific subisotype compartments driving, associated with the disease. Next slide. Given that we're working with Kevin and that his lab has done an extensive amount of work at looking at the V gene association with myasthenia gravis, we want to make sure we can recapitulate that data and see if our method could add any further resolution. Hit the button again. What we did see, thank you very much, is the association, statistically significant association of these V gene families, V2, V3, V5, V6, and they range, and then we broke it down by sub-isotype, right? So in V2, you see a statistically different association between healthy and myasthenia gravis in the IgG1 compartment. In the V3 gene family, it was also an IgG1. IgG5 was in V1, V6 was in 1 and 3, and V7 was in 3 and 4. But when we wanted to look and see if there was an association in the different distribution of the constant regions themselves, so we know, for example, that IgG1 and G2 are the highest in the circulation, and 3 and 4 are the lowest, but they still can show differences between sick and healthy. We did see only in IgG3 was there an association of higher expression of IgG3 amongst the B cell repertoires than in the healthy donors. So that suggests that IgG3 is expanding and that it's doing something here. Next slide. Oh, yep, there you go. Next one, please. So again, we went through and looked at these individuals. I just want to show you again what some of these plots look like. This is a small subsection. Putting a tile plot with all these colors for 96 individuals to be a little overwhelming. So I'm just showing you 7 MG individuals here in this tile plot and 4 healthy donors. The novel alleles are marked with FL or stars on the right-hand side with a color. One more advance. So we can see, again, these IgG4 duplications in these alleles represented by 3 alleles being expressed in this individual, not 2. Now, I will say a caveat here is that if you have a duplication, but that duplicated allele is the same as the one you're upstream, we wouldn't see it here, right? We'd only see it based on sequencing differences. So this is the sort of bottom, the floor of how many duplications are in this cohort. Next animation, please. Overall, if you look at the unique alleles, you can press it one more time. In this cohort, we see that it's represented by these stacked bar graphs, that the purple boxes represent the alleles that we identified that had already been identified in IMGT, and the blue represent alleles that were novel and not in IMGT. Can you animate one more step, please? So in the healthy donors, 63% of the novel alleles we found across all 4 sub-isotypes were novel, and 79% of the alleles we identified in the MG cohort were novel. So the vast majority across all donors were novel compared to those represented in IMGT. So this not only suggests that our resources are under-sourced, but also that there may be novel alleles in here that could help our identification of potential biomarkers or risk factors in MG. I think I do want to point out something that is really unique, not specific to MG, but something that these resources can help, is that, you know, a lot of our discussion about understanding antibody evolution, you know, discussed in the previous talk and in others, is looking at the rates of somatic hypermutation and the extent of mutation away from the inferred germline using AirSeq data. If the databases themselves are not complete, then what you're inferring against is an incomplete database, and you actually may mark something as a mutation when it actually represents a novel germline allele. And so I think this is also just a highlight and an overarching call for more genomic resources for immune repertoires in general. Can you go to the next slide? So our current efforts, you can click one more, is really looking into these constant region functional signatures. There is excessive evidence in the literature, a lot from the antibody engineering field, that have identified very specific residues in the constant region in IgG that are responsible for receptor avidity and affinity, for interaction with C1q for complement, fixation for glycan-mediated changes that impact function. And so we want to look for the alleles that we've identified with the knowledge that's out there to see if we can see any of these patterns that may suggest what these new novel alleles that we're identifying in our disease cohort may be doing. Next slide. So a great example of this is this hinge deletion. So this is an alignment of IgHG3, the IgHG3 constant region. On the left are the different alleles we have aligned. These ones that are starred on the bottom are 5 of the new ones that we identified. And we do see a distribution of deletions of hinge 3, the alignment on the top showing the organization of this constant region chain in the alleles, both in the database and in the novel ones that we discovered. And there is older papers out there from the 90s, and even more frequently or recently, from SARS-CoV-2, that have identified that IgHG3 hinge deletions are associated with more robust complement activation and potentially higher pathogenesis in the context of vaccination or disease. Next slide. So we wanted to go through and look at this hinge deletion in particular, just as an example of an added amount of data that we can bring with higher resolution of the MG cohort. So we're looking for the difference between this M allele, the major allele that has these 4 hinge copies in IgG3, and the S allele, which will have these 3 copies. Next animation. So in parallel with this MG project, we've been continuing our healthy donor population-based sequencing, and this graph represents the distribution of these alleles across healthy donors 105 healthy donors across major global subpopulations. And what you see is there is natural variation. So the color coding here shows whether you have the 2 major alleles as your maternal and paternal haplotype, that would be green. If you have 1 major and 1 short, that would be red. And if you have 2 shorts, that's blue. And so what we see in our healthy cohort is that there is a small portion of individuals, healthy individuals in African populations and in Southeast Asian populations that do have evidence of some of these shorter alleles sort of floating around naturally in the population. If you can advance the slide. But if you look at the distribution of these alleles in our cohort, right, so this again, not incredibly powered, just 48 individuals in this group, and these individuals were not segregated based on their pathogenic mechanism or any functional data, we do see the carriage of some structural variants in the healthy donors, but more of these structural variants in the MG individuals. We also see that the MG group has 2 individuals that have this duplication of IgG3. I referred to this earlier with IgG4, but if you can advance one more time, we do see, this is a schematic of this, the references on the bottom, all green boxes, and the structural variant we're representing here is a duplication of IgG3 as well as a duplication of IgG4. And a lot of these individuals have both duplications in their genome. And so we only see this additional carriage of an additional IgG3 allele expression in the myasthenia gravis group, and that some of these additional duplications are the short allele compared to the major. Next slide. Just because we see it in the DNA doesn't mean it gets into the repertoire, so we wanted to make sure that was the case. So we went into our FLAIR-seq data, which is on the bottom, mapped it to that deleted allele from the DNA capture. So on the top, you see those 2 solid gray bars. The bottom one has that 178 base pair deletion. That's your hinge 3 deletion. And we do see it then in the RNA data on the bottom. We do see it's missing in that bottom box, which is that second allele that has the hinge deletion. Next slide. So our next steps then are to work very closely with Dr. O'Connor's lab to understand if this deletion actually leads to different functional analysis. The deletion is just a very clear example of what we can see, but we're also coming through the novel alleles we found in the MG cohort and what else is known about residue changes that change other activity, complement activity, ADCC activity, FC binding activity, to identify a panel of variants that we want to test functionally with the cell-based assays that Dr. O'Connor has stood up in his lab that was just discussed in the prior talk. So in this case, you go to the next animation, we would then plan to clone these alleles into the constant region, which again is sort of the reverse of what we normally do to characterize antibodies, of course, using a few different matched VDJ variable regions, just to make sure that as we were discussing, the angle of the variable region doesn't impact the activity of the functional domain, to really understand what the FC activity is and how it's affected by these genomics. So next slide. So overall, these repertoires are more clonally expanded than that of healthy donors at both the isotype and subisotype level. IgH3 usage is higher in MG repertoire, and we do see V gene bias, which aligns us with the previous literature. Next one. 79% of CG region alleles identified in the myasthenia gravis subjects were novel. I think this is an incredibly important point, not only to represent the dearth of reference information that we have, but also the possibility of using these as immunogenomic biomarkers. Hinge length variants were more frequent in MG individuals and healthy donors. Our goal is to use this data to possibly identify immunogenomic biomarkers for pathogenic mechanism and or risk. Clearly will require a more well-controlled and larger follow-up study, but I think the data is very exciting for now. And what this means that we can propose, if we take some more time, is a model, right, where if you are one of the individuals that unfortunately carries an autoreactive VDJ that pairs with an IgG3 hinge variant, which clonally expands, right, there's a lot of ifs, that may lead to a complement activating pathogenic antibody, as opposed to just carrying an autoreactive VDJ, which may act through the 2 other mechanisms. So we can go to the next slide. I really want to make sure I thank my group and our collaborators at Yale, specifically Easton Ford, who's on the call here and is in the middle of that picture. He did all of this work for his PhD thesis. There's a QR code there if you're more interested in the FLAIR-seq assay. I think there's a lot of results that you guys are talking about today that could benefit with this linkage of the constant region and variable region that the assay could provide. And with that, I'd be delighted to answer questions and further the discussion.
All right. Thank you so much. And questions from the audience?
Or not, and we could rescue time for lunch.
No. So I have a question here. So how far are you from actually being able to apply this immunogenetic screening to patients? And maybe not you specifically to myasthenia, but in other autoimmune diseases or responses to vaccinations or, what have you.
Yep. So we actually have a very demonstrable proof in the context of HIV germline targeted vaccines, influenza vaccines, and COVID vaccines of distinct germline alleles that influence response. The HIV vaccine is a very strong example because it's designed to only target one V gene allele. And so if you don't carry that allele either through deletion or levothyroxine, you won't respond as intended. And so we have some pretty amazing data that shows that over 70% of Southeast Asians actually don't carry that allele. And it's something that we're actively in discussions with those folks at Moderna and Scripps who are running that phase 3 clinical trial currently. In their phase 1 trial for the VRC01-like immunogen for that vaccine, they tested the vaccine in 40 individuals. They only had one person who didn't respond, but that one person's non-response was completely attributable to their antibody allele. And so I think that we are very close to putting this into practice for screening in the context of vaccination. I'm really excited about the possibility of autoimmunities, and there's so many B-cell autoimmunities. MG was a very easy place for us to start because of this added feature of being able to separate the subisotypes. Because IgG1 and 2 are so dominant in this era, being able to really hone in using sequencing data on just the IgG3 compartment and which VDJs are being associated with which IgG3 alleles was an obvious sort of value gain by using our assay in this disease context. But I'm really interested, my passion is understanding the functional implications of these changes. And so given what's already known about the pathogenic mechanism, I'm really excited to expand upon these data in MG in particular and try to understand if that's a possibility. But the test as a screen is already ongoing in other disease models.
Cool. Dr. Kushner.
So just to kind of understand this, if a patient has like an IgG3 allele that would produce a stronger response in complement,
right?
Would that suggest that any antibody that they are producing would be a stronger IgG3?
Right.
Or is it just specific to acetylcholine receptor?
No, I think it does indicate. I mean, it depends on the heterozygosity of that allele, right? If they have two alleles, the maternal and paternal allele that both have that deletion, all of their IgG3 antibodies, regardless of the specificity, would have that potential, right? But that needs to also be paired with an autoreactive VDJ. And so I think in that case, any autoreactive VDJ with a IgG3 deleted hinge deletion allele would have more potential. And this is where we really have to dig into those functional assays because the data that's out there demonstrates that not every antibody only acts through a single mechanism, right? That they can do multiple mechanisms. And so we need to understand how much of the mechanism is driven by just this constant region, which is why we're really excited about the idea of partnering with Dr. O'Connor to do the sort of paired functional testing where the VDJs are matched, but only the constant regions are changed. But yes, if they have that constant regional allele that has that deletion, and that deletion is truly associated with increased complement activity, then any IgG3 that uses that allele would have the potential of that activity. Now, there's all kinds of questions about B cell selection and allelic exclusion and class recombination and the way that happens and the folding of the DNA. Whether we know at least, right, that it makes it into the RNA. So the next step beyond function is also to make sure it makes it into the sera. And so we're doing those proteomics experiments right now.
Yeah, just to take it further, so there's a lot of ideas of like the autoimmune disease itself tends to break off into other autoimmune diseases.
Right.
So does that play into this theory of like these alleles and the product of producing these antibodies that are more pathogenic?
Yeah, if I were to speculate, I would say absolutely. I think the other thing that we're really interested in, and I have someone in the lab, actually, he's here in the picture. He's the one with copious tattoos. Probably the most incongruous person who's working on estrogen response in IGH. So are there differences in, estrogen stimulating specific classes of antibodies that then move on to stimulate specific autoimmune diseases? So I think there's a lot still left to be explored in this regard, and why autoimmunities are associated with each other, and why autoimmunities may be associated with different hormonal profiles as well.
Excellent. Anybody else? So we're actually out of time, but I don't care. I'm going to ask this question. So you still have relatively small numbers, right? And speaking as a clinician, ultimately thinking about the value for this for me, right?
Right.
And you've got to have high specificity, sensitivity, and more importantly, positive predictive value. So do you have an idea what kind of N you want for the number of DNA samples? And can the MGNet and other people on the line who have PBMCs that are just hanging out frozen,
Yeah,
would you be interested in those?
Yeah, I think that we certainly would be interested in those, and I think that is something that we're in the process of calculating. We just did, a lot of this data was initially done using the RNA assay. It's cheaper, faster, all of those things that we're now confirming. We're just validating everything with DNA as well. And then once we have those two numbers, they're validated, all of our structural variants, as I outlined them today, look solid, then we're going to be calculating those types of doing those types of power calculations, identifying what types of numbers those are, and really trying to work with Dr. O'Connor to seek funding to support that bigger expanded study, and then maybe also figuring out what maybe other sort of partner or associated autoimmunity we could look into as well. And then, yes, absolutely. I think, samples are always the most primary clinical samples are always the most limited resource. So anyone that's willing to help, I'd be delighted to hear from. I think one of the things we wanted to do but couldn't do, again, for the small study size, is to associate these alleles with stage. And so, again, a larger cohort will allow us to do that as well. So I think there's a lot to be said for more samples moving forward.
All right. Sounds great. Thank you.
Thank you.
So we are going to end the morning session. We'll be back at 12:30. And so see you then.